So let's move to the second stage of protein synthesis, the initiation, the most important step in translation reaction, because this is the stage where you can control the translation event. This is the site where the most of the regulation is happening. Okay, so in the first stage we have seen that an activated amino acid is charged onto a tRNA to form an amino acyl tRNA. In this stage, or the initiation stage, all the four most significant components of translation will be gathered together. That means the ribosome, the mRNA, the tRNA with the amino acid will be gathered together to initiate. Okay, so we will describe the initiation stage in 3 steps. Okay, step 1, step 2 and step 3. In the step 1, there is the 30th ribosomal subunit will interact with the mRNA, and in the step 2, the tRNA will be recruited into the 30th plus mRNA complex, and in the third step, the 50th will come into the already existing complex to form the initiation complex. Clear? So it completes in 3 steps, right? So actually what's happening in the initiation is there is a complex formation between the 30th ribosomal subunit, the mRNA, the FMET tRNA FMET, and the 50th subunit. Additionally, we have to spend GTP molecule. Actually, one molecule of GTP will be spent over here, and there are 3 initiation factors. That means there are 3 external proteins also involved in the initiation reaction. They are called initiation factors. It's shown there. You can see this is the IF1 or the initiation factor 1. Their corresponding function is to bind to the A site of the ribosome. So what is this A site? Within the ribosome, we have 3 sites for binding by the tRNA molecule. They are denoted as E, P and A. We will see that in the coming sessions. There are E stands for exit site, P stands for peptidyl site, and A stands for amino acyl site, right? So the IF1 and within the ribosome, we have, as we have 30th and 50th subunit, these E, P and A sites are usually present in both the subunits. That means in both 50th and 30th we can see P site and A site, but in case of E site, most of the part of the E site are residing in the large subunit. That means in the 50th alone. Okay, and the mRNA usually interact with the 30th subunit alone. Usually there is a small entry point, or there is a small orifice within the 30th subunit through which the mRNA can pass. Okay, so back to the initiation factors. There is IF1 which binds to the A site and which block the tRNA attachment during initiation. Also, not only the tRNA attachment to the A site, but also the other factors to the A site. That means A site has to be protected during initiation without binding by factors or the tRNA molecule. And the IF2 forms a complex with the tRNA. That means the FMET tRNA FMET, and that will be recruited to the middle site called the P site, right? And IF3 usually will bind on to the 30th subunit and prevent a premature assembly of the 50th subunit. Okay. Now the step 1 initiation, in which first the 30th subunit will be bound by the 2 initiation factors. That means the IF1 will bind onto the A site and IF3 will bind onto the 30th subunit, and the functions already we have seen. And now in the second step, the mRNA molecule after transcription will be bound onto the 30th subunit. And as we know that in prokaryotes, once the transcription is being going on, the 5 prime end of the RNA will be coming out of the transcription complex, right? And to this 5 prime end, after the 5 prime capping, or after this mRNA is being synthesized, this ribosome will occupy the 5 prime end of the mRNA molecule. And the ribosome means here it is clearly says that it is the 30th subunit. The first subunit of the ribosome attaching to the mRNA will be the 30th subunit. Clear? And the initiating sequence, that means AUG is the start codon. So this codon within the mRNA will be guided to the P site, and this is the site where the AUG has to occupy in order to start in translation. And this is guided by a particular sequence in the mRNA called Shine-Dalgarno sequence. Clear? So this is the first step. There is IF1 and IF3 binding onto the 30th subunit, and also the mRNA is attaching to the 30th subunit. And the attachment is precise, has to be precise to position the AUG at the P site. And this is controlled by an internal sequence of the mRNA called Shine-Dalgarno sequence. And what is this? SD sequence or Shine-Dalgarno sequence. It's called because it is named because of the scientists who discovered, right? And actually this is a consensus sequence of 4 to 9 purine residues. You can see that in the image within the mRNA, usually located 8 to 13 base pair upstream to the start site. That means upstream or 5 prime end of the AUG codon. And what is the significance? As already said, this helps the AUG codon to be positioned in the P site. That is the most significant one. What are the other significant features? This sequence base pair, or how this AUG is positioned exactly at P site. The mechanism is this sequence base pair with a complementary pyrimidine rich sequence within the 16S rRNA of the 30th ribosomal subunit. So we have to recall that rRNA are present in the ribosomes, isn't it? So within the 30th ribosomal subunit, we have the 16S rRNA. So within this 16S rRNA, towards the 3 prime end, there are certain sequences which are pyrimidine rich and are complementary to the sequences at the Shine-Dalgarno site. Clear? So there is an mRNA rRNA hybrid formation, and this is actually helping to position the AUG to the P site. Okay. And again, due to the SD sequence, which helps to distinguish the AUG for binding by the FMET tRNA FMET from other AUG codons within the mRNA. What is it? Which means that we know within an mRNA we will have AUG at the start as a start codon, and also we can also see AUG codon at internal positions of the mRNA. So how the tRNA, that means how the tRNA FMET tRNA FMET identifies that this is the first start codon, and to that site I have to go and attach. So how the tRNA recognize this? This recognition is done through the presence of a nearby Shine-Dalgarno sequence. So to the AUG, which is actually at the start site, the tRNA carrying the formyl methionine will be attached only if there is a nearby Shine-Dalgarno sequence. Clear? So this sequence is important. In eukaryotes, we have a different type of sequence, what we call the Cossack sequence. But in prokaryotes, the sequence is called the Shine-Dalgarno sequence. Okay. Now the second step of initiation. Here, the FMET tRNA FMET has to be recruited into the P site. Clear? Normally, the FMET tRNA FMET will have an anticodon sequence UAC, which will be complementary to the AUG codon at the start site in the P site of the 30th ribosomal subunit. Okay, so in the first step, this FMET tRNA FMET has to be recruited into the P site, and this is supported by an initiation factor called IF2, and this IF2 is nothing but a G protein which can bind GTP and can use this energy from GTP, right? It has GTPase activity also. So this IF2 GTP will combine with the FMET tRNA FMET, and this complex, that means the FMET tRNA FMET IF2 GTP complex is actually being occupied in the P site, right? And as a second step, the codon anticodon will interact further to form a complementary pairing, and that holds the tRNA precisely in the P site. This is the second step of initiation. Clear? So there is the complex tRNA FMET tRNA complexed with IF2 GTP will be positioned into the P site. And in the third stage, or in the third step of initiation, the 50th ribosomal subunit is coming to bind onto the counterpart, that means to the 30th subunit. And binding of the 50th subunit will cause some conformational change, and this conformational change causes the hydrolysis of GTP bound to the IF2 and to the tRNA, and there will be release of GDP and pyrophosphates, right? And not only the GDP will be released, but also the other initiation factors. Actually, the IF3 will be released even before that on attachment of the FMET tRNA FMET GTP IF2 complex attachment itself, and following the hydrolysis of GTP, the IF3 will be released. Then there will be the 50th subunit coming into the complex, and when 50th is attached onto this, IF1 and IF2 also will be expelled out of the complex, right? So now an active functional initiation complex has been formed, and this is called initiation complex formation, and then it is ready for the addition of next amino acid into the next site.